listen to this after this session. Okay, so the first slide is not sent to you because it's just a general slide to help understand the general structure of biosensors as well as bioreceptor. I hope, Sue, can you help to accept the participants? Sometimes they have internet connection, so they sort of join
okay,
in and all right. Okay, so
okay.
all right, so this is the all right, so okay. All right, so the first thing that we are going to look at is the basic structure of biosensors. And these are, we have a lot of experts here, but just to introduce to those who are not familiar. So there are many things is important in the basic of biosensor, including lateral flow is considered a biosensor. But the one thing that really what we call as the heart of biosensor is the bioreceptor. So we cannot just simply take any bioreceptors that we think suitable. It has to be studied, it has to be characterized, and the binding, especially the binding, the selectivity has to be done. The study has to be done before the construction of biosensors. And of course, in lateral flow, we are going to discuss about the bioreceptor part in the second part, as well as what type of analyte suitable for lateral flow. And the transducer is the lateral flow parts, and some lateral flow also has a special reader. So now we can attach, we can use, especially our mobile phone now, to have, to help us to identify the signal readout from the lateral flow. So the interaction that happens between the analytes and the bioreceptor is the one that we have to study before the construction of lateral flow. If the interaction is optimized and confirmed, then we can do the construction of lateral flow. Without that, I think if we just straight away embed the bioreceptor on the lateral flow without confirming the interaction, then it will be a very long journey. So it has to be confirmed first and then optimized, which is not in the lecture, which is not will be in this lecture, because it has to be done via standard techniques, okay, using SPR, using ITC, okay, which is not discussed in this lecture. So for those of you who are not familiar, okay, we have some common, okay, common used bioreceptors, okay. So we have aptamers, we have antibody, and we have DNA. So these are the different in terms of general structure of antibody, aptamer, and DNA. So important to know, because for us to conjugate, yeah, later we need to conjugate with the nanoparticle. So we need to know the structures, which part that we are going to attach to the nanomaterial. So there are two parts that you need to know. One is the chemical, the functional groups of the structure, the bioreceptor structure. For example, antibody. I'm going to show you, okay, the DNA structure is quite common because you some of you are even more expert. Okay, just a moment. I'm going to skip to. Okay, so some of you are probably familiar, okay? So because commonly used in lateral flow is antibody sets. So we need to know about the functional groups that exist in the antibody. So we have carboxyl and we have amine. So to make sure that the antibody is clearly exposed to the antigen, it comes to the antigen. Okay, I hope that I can tell you first about the difference between antigen and antibody. So this is the antigen. The antigen are the one that we are exposing to, or the one that we wish to measure. Sometimes in chemistry, we call it analytes, but in biochemistry, we call it antigen. The virus, the bacteria, those are the ones that we want to detect, is called antigen. And you can see that the antibody, okay, the red one, is attaching itself to the surface of the antigen, and we call it epitopes. Yeah? I hope everybody can mute first. This is just an introduction first, all right? Okay, now, so I hope everybody can be clear on the functional group, because if you fail to put the right functional group on top of the lateral flow or the sensor platform, then you might not be getting a good signal. Okay, so that is the most important, because we want the carboxyl part to be on top of the lateral flow, not the amine, because this is where the antigen interact with the antibody. Okay, so that is for those who cannot or do not have are not familiar with the basic of biosensors. Yeah? For those who are an expert, please bear with me first. And then, of course, we have aptamer. And this is a very good alternative. We have also used this in our applications, but we haven't used this. Our group has not used this in lateral flow yet. Okay, I think soon we are going to use it in aptamer, but we have done this using electrochemical technique. Okay, so we can also use DNA for lateral flow, and bear in mind that you have to be very careful on what kind of linker that you wish to have, so that you can attach it with the nanomaterial. And the nanomaterial as well, if you have to be careful what chemistry that you supposed to have. For example, if you want to attach it with gold nanoparticles, then you need to have proper thiol, for example. You can also use silver nanoparticles. Okay, we can discuss this in the second part. And also, if you wish to have silicon surface, then you have to make sure that the chemistry of the linker suits the nanomaterials. Okay, so if you choose quantum dots as your nanomaterials, then you make sure that the materials of the quantum dots or carbon dots, for example, suits the chemistry of the linker for your bioreceptors. Okay, so this one is just to introduce to those who are not familiar with the basic of the bioreceptor. So now we are going to move to the lateral flow. So let me switch to the new slides. I hope that everybody can mute first, and then once you have, if you have any question, you can stop me, definitely. Okay, right. So this is the basic, the first part will be the basic of the lateral flow, and the date is not correct, it's 5 to 6 July. So today is 5th of July, okay? So the basic in lateral flow, we start with, of course, the introduction of what is lateral flow immunoassay. And of course, it is a popular technique because it's a rapid test, not expensive. I think I have to check. All right. Okay. And probably many of us don't know that it's not just blood, it's not just saliva, but it can be used for many types of samples. Okay. Even you can have food samples, blood samples, brain samples here. Depends on how you prepare the pre-treatment of the samples. So it can be done. So test sensitivity can be quite good, unfortunately, for lateral flow. It is quite what we call as not compatible to a good technique, like benchtop analysis. Or for example, we have optical techniques, spectrophotometer, fluorescence meter. We cannot have that compatible sensitivity to the benchtop technique, but it's a rapid test. It's a good rapid test. But now we can enhance the lateral flow so that we can improve the sensitivity. There's a lot of ways that we can do to improve the sensitivity, but generally lateral flow is not sensitive. Okay, so previously we are very familiar in using lateral flow for COVID tests because COVID is virus. So in terms of viral load is quite high. So that's why we managed to get reading, easy reading, testing virus. But then if we have like bacteria or a very low concentration of antigen or analytes, then it can be difficult to get a good reading using lateral flow. Okay, so this is the basic structure. I'm sure that many of you are very familiar with. This is the basic structure of lateral flow strip. Okay, so bear in mind we have to focus on the sample pad because this is where you put the samples, regardless if it is blood samples, if it is saliva, sputum, urine, or any solid samples that you homogenize first. This is where you introduce. And sometimes we call it backing pad, sometimes we call it wicking pad. There are many names for the backing pad. And the conjugate pad is where we put the conjugated nanoparticles, okay? And of course, we have the test line and control line. And this is the last part, which absorb the excess fluid moving from here to here. Okay, so this is the basic structure. And for in detail, we have to be very careful about the bioreceptors. And in this case, I'm referring this as the detection bioreceptor. And this is capture bioreceptors. So in using antibody, you have to be sure which is which. For example, this one, the detection antibody, usually we prefer to use monoclonal. And for the second one, which is the capture antibody, it is suggested to use polyclonal. So for monoclonal, of course, it is much more expensive. So we have to optimize. And this capture or this test line requires higher concentration compared to the conjugate pad where the detection antibody will be placed. So this one requires higher concentration compared to this conjugate pad where we put the conjugated antibody. Okay, so this is the basic one, what the lateral flow looks like. And of course, this is also some basics that you know where you get positive results, okay, and the negative results. Okay, the structure of where you get this is the test line and this is control line. So if it is negative, you get only control line, whereas when you have positive, you have the both lines appear. Okay. So this is basically how to control, yeah, the sensitivity as well as whether you are going to get a good results or you're going to get difficult to observe. Okay, one, of course, what we experience is the solid support itself. Okay, so if you don't have a very good, because if you buy from the suppliers, we have to check. Okay, there are certain characteristics of the nitrocellulose membrane. Okay, the solid support. If you don't buy a good one, then you have difficulty in having the conjugated antibody just now. Okay, the conjugated antibody to move, okay, to the wicking pad or the absorbent pad, because this membrane, okay, plays a big role. Okay, if it is like if it moves too fast, then you cannot have a good interaction here. If it moves too slow, then you might not be getting reading at all. So the quality of the nanocellulose membrane, so the nitrocellulose membrane is very important. And this thing should be discussed with the company which is which. Okay, they have their own characteristic. So the commonly used membrane is nitrocellulose, where there are also some other types like nylon, what we call as glass fibers. But of course, those quite maybe expensive. So this is the best option. And you have to check on the porosity as well as what kind of the one that is offered by the company. So of course, advantages of nitrocellulose, low cost, okay, capillary, okay, the flow characteristic is very important, and ease of handling. But sometimes when it is wet, okay, it tends to swell. So especially for those samples that we prepare in a lot of liquids, then we have to be very careful to check on whether it is suitable or not to be used with the correct nitrocellulose membrane. So this one, I think, because what we have experienced is not to buy many. You need to test on a few examples of nitrocellulose, and then you choose the correct one. All right. We have hyaluronidine. Okay. This plays a big role actually, apart from the bioreceptors, but the structure or the solid support of the lateral flow is very important, okay, to choose. This is based on our experience as well as based on my reading, okay? So this is where conjugate pad is where we put the conjugated nanoparticles, okay? So of course, as I mentioned before, commonly used is gold nanoparticles, but instead we can use quite a number of different types of gold nanoparticles, not gold. Apart from gold, we can use silver, carbon dots, dye, conjugated liposomes, magnetic nanoparticles, okay? Depends on what your application is, okay, right? But of course, it has different costs in terms of fabrication, and gold, of course, is quite common and reasonable price, okay? And of course, the nanomaterial has to be conjugated first with the antibody, okay, before it is embedded or casted on the conjugate pad. We will discuss the detail of how the condition of when we need to expose the conjugated antibody or gold nanoparticles or nanoparticles in the second part, okay? So the color of the nanoparticle, okay, must be visible, okay? If you are using carbon dots or quantum dots, it is unlikely that you can observe using naked eye, okay? It requires an external reader. You have to develop your own reader so that you can expose the carbon dots, okay, to UV light before you can read the test line. But if it is gold and silver nanoparticles, the color is intense and you can just observe the lines by naked eye. And of course, by using this technique, for example, the gold and the silver nanoparticles, they are not that sensitive. Okay, sensitive in this case, they are not responsive to low concentration of antigen or low concentration of analyte. Okay, but using carbon dots or probably quantum dots, because theoretically it will be sensitive, much more sensitive, but unfortunately you have to use external reader. You need to have some sort of reader to be able to read the test line, okay, right? So the requirement is the label, means that the gold or any nanomaterial used has to be stable, okay, in liquid or dried form, because in the end you have to dry the lateral flow strip properly. We have to heat it up until 60 degrees, 37 degrees. So it must be very stable, okay, the nanomaterial as well as the antibody. And it should be non-bleaching, means that it does not leach out from the lateral flow. So those two are very important characteristic before you choose the nanoparticle as well as the bioreceptors. So commonly used is gold nanoparticles, okay? And the size, okay, that has proven to work, means that it is observable by naked eye, is around 20 to 40 nanometer. Okay, so if you use, I think based on our experience, 40 nanometer is among the best one. Okay, I think 20 is not that really useful if you want to observe by naked eye. Okay, so first thing that we have to do is to make sure that the gold nanoparticles or any nanoparticles that you wish to use in fabrication of the lateral flow, it has to be characterized first. Unfortunately, it has to be done for every batches. Because so whenever that you want to construct the lateral flow in for mass production, the nanomaterial has to be characterized every time. You cannot like, okay, this is the one that I have produced before, I'm going to reproduce, okay, but sometimes it can affect, so it's good to sample some of the newly produced nanoparticles, check the size, and then you use it, and then you use it. If not, then you might have a difficulty in getting a reproducible results. So every time you have to test the stability of the gold nanoparticles or nanoparticles that you have synthesized or you might buy from somewhere. Okay, so this is very important. If not, then the possibility of failure to get a good results comes from here, the stability of the conjugates. Okay, any mentioned here, any protein molecule can be attached to gold nanoparticle. Yes, because since gold nanoparticle as well as silver nanoparticles, they are very easy to attach to biomolecule. So when based on our experience, it does not require any linker. We can just mix together the antibody and the gold nanoparticle. We haven't done silver nanoparticle. We can just mix together the gold and the antibody together. We don't need any special linker. Okay, so we observe that this works for us. Theoretically, according to theory, we need to have a pre-treatment for the antibody to be able to anchor to the gold nanoparticle. But based on our experience, we don't need that. So I don't know, it's up to you whether you want to modify first your antibody set to have a proper linker so that you can attach to the gold nanoparticle, or you can just incubate the antibody that you're going to use, the gold nanoparticle. So this is the basic of the gold nanoparticle, and you can also use silver nanoparticle. So the sample pad, okay, this is the commonly used being as a sample pad where the sample is being introduced to, and of course you can use cellulose glass fiber. So cellulose is common. Glass fiber is expensive, but it gives a better filtration. Okay, if you need to filter your samples, so glass fiber is giving you an option to filter. So for example, if you have a blood sample to be introduced to the lateral flow, because blood is red, okay, is going to give you some issue in reading. So this glass fiber can help you to filter, to separate the red blood cell with the serum. So there are options. We never use this because it's quite pricey. So if you wish to have this, you can discuss with the company. So probably they have options for you. okay? But we commonly use cellulose. And of course, during the production, which will be in part 2, we need to use certain buffer, okay? Surfactants, blocky reagents, in order to avoid non-specific adsorption or non-specific binding, as well as to increase sensitivity. So this related to pH, related to ionic strength of the solution. So this one comes in the later part, okay? And of course, the last one is the wicking pad. The last is the wicking pad, means this one. This is to pull, right? The sample or whatever reactions to go towards the end. And it stops the whatever liquids that has moved forward from moving backward. Okay, so we do not want the whole lateral flow to swell. Okay, so that is why we need to make sure that the wicking pad is working, is functioning well. If not, then we do not want the fluid to be released back. Okay, so the thing about this is we have to test and whether the lateral flow is working or not. Sometimes the storage plays a big role, because if we don't put the whole strips or lateral flow in a proper storage condition, then you might have issues with the reading as well. Okay, so backing pad is just the support here. Is where you put everything, all the sample pad, conjugate pad, membrane, as well as the wicking pad is where you put on the as a support. So as long as it is strong, then it's good enough, and easy to cut, because in the end you have to cut the whole thing to a proper size. So that is what backing pad or is the function of the backing pad. Okay, right. This is the process of labeling, as I mentioned just now. So I think we are going to discuss this in the second part of lecture. So make sure that you have identified the correct biomolecules that you wish to detect. Okay, bioreceptors, the suitable bioreceptors, okay, and has to be done before the process of fabrication of lateral flow started. It has to be done before that. So basically, what should be in the test kit using lateral flow. This is an example of rabies test kit. This is antigen test kit, and you should have a proper buffer, okay? And this one, I think this is quite common that we have in a test kit. So we have the strip itself, okay? And of course, the swabs, okay, to take the samples and droppers. That depends on what types of applications that you want to use the lateral flow for. And it is very important. For example, this is based on our experience. If any of you wish to commercialize these types of test strip, you have to put what we call as precaution. I don't know whether that is the right word. Okay. For example, if you wish to test saliva, then you have to put instruction that before the sample is taken, means it's saliva. Okay, the particular person has to not taking any food, clean their mouth. Okay, those are precautions that are very important and is very crucial during the sample taken from the saliva. So there are many things that we will be affecting because we have to think about all the proteins in the food or in the drinks, in the mouth. So you have to be careful about this, because if not, then people might claim that your strip is not working, but you have to think of all the possibilities that will happen if the samples were not taken properly. So we have to think about the precautions. So if you're thinking about the food samples, if you're thinking about food samples, then you have to put the pre-treatment as well. You have to consider pre-treatment as well in the kits. So whether you have simple extraction, you have simple digestion of the food samples. So these are very important to be considered with. So these are basically the protocols. I'm not going to discuss this actually. So, but one thing that we experienced is when we did tuberculosis, we are working with sputum samples, okay? Since the lateral flow is not that sensitive, if we use a lot of buffer, we cannot get the reading because the sample is too diluted. But if we choose not to use buffer, the sputum, because they have different viscosity, some sputums are not too viscous, some sputum are too viscous, then we have problem getting the reading because the sputum will not move to the end of the strips, okay? So there's a lot of problems actually, and that depends on the type of samples. So if you choose to commercialize lateral flow, you have to think about the samples itself, how the sample should be. The sample should be clean, or the person should not be, for example, smoking before the samples taken, should not eat, should not drink, or should clean their mouth, should clean their nose or whatsoever. Those are very important precaution that you need to put on the kit before getting the samples into the proper buffer and into before testing. So that's very important. If using blood, then it's quite easy. You just have to filter the blood, okay? So that requires you to have a proper, what do you call? Just now I mentioned about the glass fiber. Okay, the glass fiber will help you to filter the whole blood, okay? So this will avoid the red blood cell to interfere with the reading. So this is very important, apart from the lateral flow itself, but the sample obtained, okay, is very important. If not, then we're not going to get a good results. Yeah. So these are the common ways of the protocols involved in using lateral flow as the sample, as the technique. And this is what I call as the general one, because if you're using urine, if you're using sputum, if you're using saliva, if you're using solid samples, that has to be homogenized first, then that will be different, okay? So this is the one. All right, so I forgot. If you have any question, please do stop me. But I'm sure because the part that I discussed just now is quite general, and most of you are an expert. So this is a bit of scientific, a bit more scientific. the applications or some parts of the lateral flow here. Okay, so I hope that everybody can follow me in terms of there are two types of LFA assays, non-competitive sandwich test, or you can also use competitive tests. Okay, and this based on your samples. So if you intend to do, because lateral flow can also be used to test small molecules. Okay, if you have a small molecules to be detected, you should focus on competitive tests. So if you have larger molecules, like commonly what we use to test a virus or bacteria, then we can use the then we can use the sandwich test. Okay, so this is what the difference between sandwich or non-competitive with competitive. So this one is competitive test. Okay, so what's the biggest difference in competitive test? Instead of using antibody here, because normal sandwich, they are using antibody, we use the antigen itself. Okay, my pen is not. Okay, or the samples, the analyte and the antigen, or the analyte itself as the bioreceptor. And of course, it's quite difficult because sometimes we can use the analog, sorry, the spelling is wrong. The analog of the antigen, okay, to be the capture bioreceptor. So what happened is, if there is an absence of target, means that there is no positive analytes in the samples, okay? Whatever system that you use, in this case, antibody, will interact with the antigen that is already in the test line. Okay, so you are going to get two lines, okay, and this line, the test line, is going to be very intense. Means that the intensity of the line is high. Okay, that means the color is going to be very bright. Okay, but if you have the target or the antigen in the samples, there are going to be competition, competition between the immobilized or the captured bioreceptor, which is also the antigen or the analyte, or it might be the analog. Yeah, that's correct. Okay, so there will be a competition because there is also the same antigen in the sample. So this line, when it is positive, it should be less intense. So this is competitive base, and we never done this. We have never done this since it's quite difficult actually to immobilize, to do this actually, to put the antigen, to find a good analog to put on the lateral flow. But this is very good, okay, for for those who wish to do small protein, okay, because why? If it is normal antigen, okay, if it is normal antigen, means that's a normal protein, a normal virus or bacteria that you want to detect. Usually they have multi-sites that you can react with the antibody. But if you have small molecules or small protein that you wish to detect, sometimes they don't have multiple sites, they only have a single site. So in order to detect these types of molecules, small molecule protein, okay, it's better to use what we call as competitive assays. Okay, so this probably is suitable for those bulky molecules, and this one suitable for small molecules because you don't have the molecules, the protein does not have multiple sites, multiple binding sites to be able to interact with the antibody, for example, as the bioreceptor. So that depends on your applications. Yeah. So competitive assays, okay, as I mentioned, usually we use analog, okay, to be embedded here as a capture bioreceptor, because if you use the antigen itself, it's quite difficult, okay, to get the antigen. And the second type, and the key characteristic is that a higher concentration results in lower signal. So as I mentioned, if the reading is positive, means the sample is there in the analyte or the target is there in the sample, so you're gonna get a low intense test line compared to the negative samples. Okay. Competitive assays are generally used for small target. Okay, it's very useful actually if you have a small molecule targets, small proteins, because there are some diseases that you wish to detect the biomarker, but it is a small protein, small molecule protein. So competitive assay is very important. And one of the difference with the sandwich assay is the concentration of the detection bioreceptor, means that the conjugated bioreceptor as well as the capture bioreceptor, the concentration is quite low. Okay, it's not like the sandwich, or for example, this is the sandwich assay. The concentration, which is non-competitive, we have to use more than 1 milligram per ml, okay, compared to the competitive assay. So for non-competitive, we require more for the test line, for the capture bioreceptors, as well as actually for the conjugated one, the detection bioreceptors. Okay, so this is the sandwich assay and is suitable for the big size analytes, proteins, antibodies, bacteria, cells. Okay, whereas for the competitive is for small molecules. Okay, so this is a comparison between sandwich assay and competitive assay. So for sandwich assay, it's quite straightforward. This is the analyte or the antigen that you wish to detect. So if it is there, then you get the positive line, test line. So if it is not there, you not going to get the test line. But for competitive assay, if the analyte is absent, okay, you are still going to get line, and the line is even more intense. Whereas when you have a positive analyte in the samples, okay, you have line, but the line in the test line is even less intense compared to the negative sample. Okay, so you can see this is the competitive assay versus sandwich assay. So we never did competitive assay, so I don't know whether how big is the difference between the intensity of positive and negative. Okay, so this is what we have done. Sorry, before that, in part 2, we're going to discuss more detail on the sensor design, fabrication, and some optimization. And of course, before we start the part 2, I hope that you can refer to the paper by, the title of the paper is Guide to Tutorial Design and Fabrication of Nanoparticle-Based Lateral Flow Immunoassay by Professor Arben Marcocci. It's a very detailed protocols published in Nature Protocols. So the table 1 and all these are referring to those tutorial. Okay, I hope that you can refer this before we can discuss the second part. And we have to, since there are sometimes different terms, so before the second part, I hope that we can agree on certain terms. Okay, you get familiarize yourself what is sample pad, conjugate pad, and those conjugated bioreceptors here is considered as detection bioreceptor. Okay, and those who will be on the test line as well as control line consider capture bioreceptor. And this is the membrane where you are going to put the test line as well as the control line. And then, of course, this is the wicking pad, sometimes called wicking pad, wick pad, absorbent pad, where it will stop all the flows. And this is the solid support, laminated cut. Okay, so I hope that you familiarize the term first before we move on to the second part. Okay, and this is the one that we have done before. Okay, by a few of our students who work on tuberculosis. And this is the real one that we have prepared. Probably Dr. Fatin has different formulation tomorrow. So of course, you have to prepare the goal, the conjugates, and the strips, okay? So the sample pad, sorry. Okay, this one. Okay, this is how we prepared. Okay, preparation in the buffer solution as well as the drying in the oven. Okay, so you can see that drying process is very, very, very important. Okay, if you don't dry there's a series of drying process. But if you don't dry them properly, then you're not going to get the lateral flow or the strip. Okay, you're gonna get you are not going to get a fully function strip. Okay, so this is the first process, the sample pad, and then of course you move on to the conjugation pad here. Okay, where you of course put the conjugated antibody, regardless of what kind of nanoparticles that you choose to have. And then usually here we use vacuum drying. Okay. And the third one is the membrane or detection pad. Okay. Okay. Detection pad to form the detection and control lines. Okay. So this is where you, of course, the most important part is where you put the capture bioreceptor. Okay. Okay. You have to optimize the how much that we need to put there. This requires optimization. So it's not just like one formulation suits everything. No, we have to do the optimization. And the detection pad has to be dried. Okay, the process, as I mentioned just now, the drying process, a series of drying process is very, very important. If not, then you will not get a good lateral flow. If you like don't do this drying properly, it will form what we call as syrupy solution, and it will not move. It will not move to through the lateral flow. Then you will not get any reading. So the drying process is very important. And then after all the pads, the sample pad, the conjugation pad, and detection pads are ready. So now they are embedded on the laminated or the support, the solid support. And this length is up to you actually. Because it depends also on the sample. Okay. If you think that the sample does not have enough time to flow and to interact, then you have to allow probably different length. Okay, so this is also has to be optimized actually. This is based on our experience, and so this is what the student has done. It's not 2 millimeter. This is correct me, Fatin, if I'm wrong. This is the difference between this. It's 2 centimeter, right? I don't know if Fatin is there. It's not 2 millimeter. Okay. And the next one, the synthesis of colloidal gold. So this is the one that we have used, and it works. Okay. This is very basic. What we have done. This is the reducing agent that we have used to get the colloidal gold. Right. And this is how we conjugated onto the polyclonal antibody. So as I mentioned just now, it is preferable to use monoclonal antibody for the conjugates, but it is expensive. So we use polyclonal, right? And we also have to adjust the pH of the solution before adding the antibody. This is to make sure that it does not interfere with the antibody structure or the antibody stability. Depends on the pH. And the solution is stored at 4 degrees and has to be centrifuged to remove the unconjugated antibody before embedded into the conjugate pad just now. Right? Oh, I'm sorry. Just a moment, yeah. Okay, so this is the one that we discussed just now. So this is the last one, centrifuge and to remove and unconjugated. Yeah, this is the last one. Okay, so I think this is the last one before we take a break. If you have any question, you can ask me now. But if you want to delay the question until the second session, also is fine. Yeah, any questions? Of course, we have emailed the slides to everybody. I don't know, Dr. Rashida, you didn't get the slides? So if Dr. Rashida didn't get the slides, can somebody give to her? Yeah, Dr. Fatin? Okay, dah ada. Dr. Rashida kata dah ada. Okay.
Assalamualaikum Prof.
Salam.
Saya ada soalan.
Mm-mm.
Boleh ke Prof elaborate more on the reader application tu in assisting the LFA reading?
Okay, that one will be second part, Diana.
Alright, okay. Okay.
Tapi that reader, because we haven't done any work on that, I can suggest theoretically only. I cannot discuss the instruction of how to build the reader because that's not my expertise. But because we don't have any existing working reader, we actually have a plan to do with mobile base, for example, using mobile phone, but it's not there yet. It's not ready yet. So I can just give you examples, but I cannot give you how to do it. Yeah, Dr. Diana?
Okay, Prof.
Alright.
Ada lagi soalan. Tadi Prof ada terangkan the step of preparation of the LFA. Is this the step tu memang untuk mass production memang kita kena follow ke? Based on your experience lah.
Okay, for the second part of the lecture, we're gonna have a bit detail on the production. Actually, Dr. Diana, I don't understand. Means that you want to skip some steps?
Ya, itu maksud saya. Kalau kita produce kan banyak in mass production, adakah boleh skip the step ataupun macam mana?
That depends on you, because if you skip some steps and you manage to get a good reading, reproducible results whatsoever, then it's okay. But for us, precaution is very important, because when we mass produce before, there's a lot of times that we cannot get the reading. For example, the whole batch is not functioning. So it's better to take precaution. Better to apply to all those, but depends on you. Based on our experience, memang banyak tak jadilah. Because probably our facilities is not that complete. Nanti tanya Dr. Fatin esok, then you know apa masalah dia lah. Banyak masalah untuk buat lateral flow ni. There's a lot of problems, especially if you don't do the storage very well. Somebody asked, you did mention that you soak the conjugate pad with the conjugate reagent. How do you control if the amount of conjugates absorbed, or do you rely on the saturation of the pad? Effendi, I think you are familiar with the process itself. Okay, memang you buat lah, Effendi. Okay. So we have we actually have done manually one. The other one, Dr. Fatin, can you answer the question for me? She's the one who do the hands-on. Dr. Fatin ada tak kat sini?
Ya, ada Prof.
So kalau tengok soalan ni, Dr. Fatin lebih sesuai untuk menjawab lah.
For manual one, we do rely on the saturation of the pad lah. Usually we use around 2 ml for one
tak dengar Fatin
pad. But for spray,
Fatin putus-putus lah Fatin.
Can you hear me? Hello?
Okay. Yeah.
Huh?
Boleh dengar.
For manual, we do rely on the saturation of the pad lah. Usually we use 2 mil, and then we air dry. And we also try using the spray method. For spray, we also use 2 mil.
I think how do you control if the amount of conjugates, soalan dia tu, how do you control if the amount of conjugate absorb onto the pad? Means that how much, is it Effendi? How much?
And we also do, because the conjugate pad is not really is not really absorbable. It does not absorb well, so we usually do a pre-treatment on the conjugate pad, so it will absorb our conjugate well after that.
Okay, I think they're using the machine. So if you use spray, you can control the dispensing speed. If using machine, okay. Susah sikit saya nak faham soalan ni. Kalau tadi kita guna 2 mil, Effendi. So this one means that saya pun tak berapa faham. Effendi, are you going to join the hands-on tomorrow? Not joining, okay. Alright. So benda-benda yang hands-on ni, I think lebih bagus dengan Dr. Fatin lah. This one, I presented some of the fundamentals and theories that I understand based on our experience lah. But I think you can, for this type of question, is okay if you can just talk to Dr. Fatin. Effendi, okay? Ini memang betul-betul dia punya proses lah. Okay. Any more questions before we stop for break? Oh, I should stop. Pause. Right. So the second part will be not to say a detailed characterization, but something that that you have to do, the system that you have to choose before you can get a good lateral flow assay system. Okay, so along the lecture as well, we are going to refer to the paper, as I mentioned, by the Nature Protocols. So please do have it ready with you so that we can discuss the academic part clearly, okay, because I cannot have all the tables in the slides together with the slides. So I hope everybody can open the Nature Protocols paper to refer to the table 1, 2, 3, and I think the last one is 4. We are going to use 4 tables from the Nature Protocol paper. Okay, so this is basically the one that I shared with you. Okay, it's very what we call comprehensive tutorials. So of course I don't memorize all, but when I read through, there's a lot of tips which you have to work in the lab to check whether you have the right protocols, or there are tips, of course, it's just tips, where it might be working for you, it might not be working for you. Okay, so that depends on how you work it out in the lab. Okay, so same terms that we are going to use. I hope that by now you have familiarized yourself with the common terms for the lateral flow structure. as well as the bioreceptors. Okay, so this is the common structures. And again, this is very important for you to master the sample types, because before you plan to do the lateral flow kit or the strip itself, you have to make sure what types of sample that you wish to for the application for. Yeah, because you can do many types of samples, blood, plasma, solid samples. Okay, so some may require pre-treatment, because if you have solid samples, we have to homogenize, we have to filter. So that is understood. And sometimes if you're using blood, okay, you must think about the separation, okay, the filtration system. So if you do this, then we have a complete solution for the rapid test, okay. And the buffer solution also has to be studied, because it cannot just be one buffer suits all. We have to try which one is suitable for your bioreceptor as well as suits the sample. So these are very important to be studied. Yeah. Okay, so that's why, because I think this is what we call as the heart of the lateral flow, the bioreceptor selection. You cannot, as I mentioned during the first part of the lecture, we cannot just jump to which bioreceptor, which biomarker, which antibody set. You have to do a lot of optimization. Okay, you cannot just buy and put it on the lateral flow, put it on the strip. Yeah, you have to study the binding affinity. And this requires you to do some standard technique analysis. For example, this is quite expensive, SPR, surface plasma resonance, or you can have option, a cheaper option using ITC, isothermal titration calorimetry, or the cheapest one, ELISA. Okay, so to check on the binding affinity, okay, the selectivity, as well as how fast the association kinetic, because in lateral flow, the process is very fast. The meeting of the analyte or the target or the antigen with the detection bioreceptor, antibody, aptamer, or DNA, as well as the capture bioreceptor is very fast. It's within seconds. Okay, so that's why you need to study the binding affinity, how fast it binds to the biomarker. If it is too slow, then it's not suitable. So this one has to be done first before the construction of the lateral flow. And it's very important to check whether the biomarker or the bioreceptor is very stable, because when we do lateral flow, it's going to be a wet and dry cycle. We are going to impregnate with buffer, and then we have to dry in the oven, or we have to dry in a vacuum drying. So it's a wet and dry cycle. So you have to imagine that this will happen to the bioreceptors, whether you're using DNA, antibody, aptamer. So if your biomarker, if your bioreceptor cannot stand this process, then it's not going to be suitable for lateral flow. Yeah, it is different if you're using, for example, spectrophotometric technique. Most of the time you use it in a wet form. You're not going to use it in dry form, as well as electrochemical technique as well. But for lateral flow, it's going to be wet and dry cycle. So it has to able to withstand this type of condition. Okay. The how to choose, I mean not how to choose, the selection of types of antibodies when you are going to use monoclonal and whether you want to use polyclonal. So usually the detection and bioreceptor, okay, this part, it is advisable to use monoclonal. Okay, and the test line or the capture bioreceptor, as well as of course the control line, it is advisable to use polyclonal, but that depends on you. Especially we know monoclonal is quite expensive. It's quite pricey, so we ourselves use polyclonal for both. So there are reasons why monoclonal is suggested for detection by receptor, because we do not want, we want to avoid non-specific absorption here. Yeah. Whereas for this test line, we want to capture as many as whatever that is moving here, right? So that's why, and then another reason why we use polyclonal for the capture by receptor, because we need high concentration of capture to have a better reading, because strip is usually naked eye based, so that's why we need to use a lot. Yeah. For example, if it is sandwich based, just now I mentioned it has to be more than 1 milligram per mil. Yeah, 1 ppb. No hidayah. Okay. So if you use monoclonal, then of course it's going to be very pricey. Okay. All right. So amount of detection by receptor is usually higher than the capture. This one because we need to capture as many analytes, as many antigen from the sample. Okay. So this is required, but then you have to optimize. Okay, we're going to talk about that. You cannot overcrowd the conjugate pad as well as the membrane test line pad. Okay, we have to optimize the how much. Okay, it cannot be too much. It cannot be too low concentration. Those are things that we have to optimize first before the final formulation is established. So here is a good tips: monoclonal antibody for detection and polyclonal antibody for capture to avoid unwanted binding. Means that non-specific absorption on the test line. So this one I have presented just to remind you basically what is happening if you're using antibody and the epitopes. Okay, so the difference if you use polyclonal, where it is anchored to many epitopes on the surface of the antigen, whereas monoclonal has a very specific epitope on top on the surface of the antigen. Okay, I'm talking as a point of view of, as a chemist, not a biomolecular, not a molecular biologist. So I'm using a very general term. So this is what that's why the biggest difference when we are using polyclonal, which is less specific in terms of epitopes, where the antibody anchored onto the antigen, whereas monoclonal is very specific, where it is anchored to the epitope of the on the surface of the antigen. So this is the suggested strategy. Okay, so those are for Um, sorry. Those are for the suggestion of bio recognition molecule or the bioreceptor. And later I will show you examples of using aptamer and antibody. Of course, aptamer is a what we call quite trendy nowadays because aptamer is much cheaper. Right, selecting nanoparticles. So this is also very, very important. What kind of nanoparticles that you can use for the conjugation process. Okay, so this one I would like you to refer box 2, page 17 in the nature protocols. So turn to page 17 just to look at the box 2. So I think just now Effendi asking about how much. So this is a very detailed process, okay, of no, this one, okay, not referring, the other one, your question will be the other box. But this one is a procedure that you can use, page 17, box 2, how to prepare the conjugated nanoparticles. Okay, how to anchor the nanoparticles to the antibodies. Okay, so you can choose to have this, or you can choose to, probably for those who haven't tried yet, you can choose to try our protocols. So tomorrow, for those who are joining the hands-on, okay, you can probably ask what kind of formulation Dr. Fatin used. So the commonly used for naked eye means that you don't have to use any device. You can just look at using your eyes. Okay, so you have gold nanoparticles as well as silver nanoparticles. So as I mentioned, the gold nanoparticles and the silver nanoparticles production also has to be optimized, need to have a good size. So commonly we use 40 nanometer for for gold nanoparticles. For silver nanoparticles, our team have not yet used this. So you can use the existing whatever published in the in any publication, but I cannot guarantee. But this one we already have established. And for production, every batch that you produce for the gold nanoparticles, you have to characterize back, check the stability and so on before doing the lateral flow fabrication. So there are other options, fluorescent types. You can use quantum dots, carbon dots, and dye-conjugated liposomes. Okay, and this, unfortunately, is difficult to observe by using naked eye. Okay, because as we know, for fluorescent material, it requires UV light, okay, to expose to the quantum dots in order to observe the lines. However, this is much more sensitive because according to the fluorescent theory, okay, fluorescent is much more sensitive compared to the UV visible technique. So if it is visible that we choose naked eye is in the visible region, okay, whereas for the fluorescent is much more sensitive. So of course, if you choose to have quantum dots and carbon dots or any fluorescent materials, you have to think about the external or additional parts, especially the UV lamp, as well as you might have to use some reader. But of course, it is much more sensitive. And apart from electrochemical, apart from optical, okay, because we are used to optical, when we talk about lateral flow, we are used to optical signal readout, okay? But we can also use magnetic base actually, and we can also use electrochemical reader. So it means that it's a combination between lateral flow and electrochemical reader, potentiostat, for example, or portable potentiostat. It can be done, okay? It does not have always to be naked eye, as well as spectrophotometric or colorimetric, yeah? But of course, the purpose of having lateral flow, we want to focus on the naked eye detection. That's because it's the easiest, it's the simplest, and suitable for rapid tests. Okay, so this one you have to select which nanoparticles that you want to focus on. And now we move back to the same structure, which is a sample pad. Okay, just now I mentioned about the sample pad, but this one we have to know a bit of theory behind what's the usage of sample pad. Okay, so whenever that we discuss with the supplier, you need to know about the bed volume. So you want to know about the bed volume, it's actually on page, sorry, sempat minum tadi. You can refer to page 8. Yeah, what is bed volume? So bed volume is total volume of the pad times porosity. So whenever that you want to consider, you have to ask the supplier about the porosity, because that one, the specification comes with the supplier. So this is important to control the sample flow, because if, for example, the sample does not flow, then you're not going to get a reading at all. Okay, if the sample also depends on what kind of samples that you wish to test. If you wish to test something very viscous, then you have to test a few types of sample pads. Yeah? So the detail of sample pads you can read, and usually we need to discuss with the supplier. And it is important also to check on the thickness of the sample pad. Okay, this is also related to the sample flow control. We have these issues because we are working with sputum samples. As I mentioned, some sputums are less viscous, some sputums are too viscous. So we have this problem, sometimes the sample does not flow. Okay, this is a big issue in our case. There are sometimes that you need a filtration system. So you can use cellulose fiber because it offers some filtration. It will filter out some bigger molecules. For example, you need to filter red blood cells. Okay, so you can choose to have glass fiber, for example. Okay, for common that we use is cellulose fiber. It's cheaper, cost, thicker, but weaker when wet. So what does it mean? That it tends to swell sometimes. It tends to swell. If you have too much liquid in your samples, then you're gonna have the lateral flow swell, and it's not gonna work. Okay, so you have to study what kind of sample pad that suits your sample. Okay, so this one is very important to for you to optimize first. Okay, so when you need to think about the buffer, okay, because the buffer will control the pH and ionic strength. Okay, because the formation, how strong the the what we call it as interaction between the antigen, the analyte or the target in your sample with the conjugated antibody, aptamer or whatsoever, depends strongly on the pH as well as the ionic strength. So you need to use a suitable buffer. This one also has to be optimized. Usually, I don't know, we use phosphate buffer, acetate buffer, and this is the range of pH because we do not want to destroy the bioreceptor. So you make sure that it is not too acidic, it is not too alkaline. Okay. So detergent, sometimes we have to use detergent in the sample pad, okay, because we want to minimize the non-specific absorption, because this is a big issue. Of course, we want the the testing to be very selective. Okay, so how does the detergent avoid non-specific absorption? Is it will destroy a low, a weak, a weak bond. So usually if it is non-target, not the antigen, it will have a weak bond with the bioreceptor. So by introducing detergent, you can disrupt the weak bond, and therefore you can have a more specific interaction with the right target, with the right bioreceptor. So this is the common detergent that you can use for the sample pad. And this one, this is quite common, not just lateral flow, we use it in other method as well. Blocking agent commonly used is BSA. This is again to minimize the non-specific absorption. We do not want, of course, because if we are talking about complex sample, we have a lot of proteins in there. So all this negative, positive charge will interact. So by introducing blocking agent as well as detergent, we can help to remove all these unwanted bindings as well as weak bindings. Yeah. Preservative, I don't know whether is a must or not. um, it is suggested to have preservative on the sample pad buffer because if you are working with sensitive samples, okay, and the process after the sample pad buffer preparation is drying, and very important, not to dry completely, not to half dry or you want to skip the process, because drying for lateral flow is very crucial. Okay, right. Next one, conjugate pad. Just to remind you on this. Okay, now the first one that we discussed. The next one, conjugate pad. Again, we have to make sure that whatever conjugated nanoparticles, whatever biomaterials, whatever bioreceptors has to withstand, means that if it is dry and wet again, it goes back to the actual structure. If it doesn't go back to the actual structure, means the structure is disrupted or something with the wet and dry cycle, then you cannot use it. Yeah. So this one, I think what you can do is to test on the reproducibility of the signal. If you get a reproducible signal 10 times, 20 times, then you're good to go. If you test the ELISA, you can only test on the specific binding. But the structure, whether the structure can withstand the wet and dry condition cycle, you have to test it when the lateral flow is ready. Okay, so function to preserve direct dried conjugated. Imagine it is dried, dried conjugated nanoparticles with the bioreceptors on top of the conjugated with the nanoparticles. And the conjugate pad also has to be able to release, okay, the conjugated nanoparticles upon wetting. So again, common material, cellulose, glass fiber, requires buffer, of course, to release the conjugated one. Drying process is crucial. You can use hot air or vacuum drying. If it is not completely dry, the conjugated nanoparticles will not be able to move. It is called syrup-like solution. Okay, it will stuck there. It will not be able to move. Then you will not get a reading. And then, of course, the next one is the membrane pad, is where you put the test line and the control line. This is the membrane pad. Okay, so in order to have a good reading, okay, you have to make sure that the capillary effect of the membrane is good. Okay, so if it doesn't move or it moves too fast, if it moves too fast, you might not have a good interaction between the antigen or the target analyte with the capture bioreceptor. If it moves too slow, it might stuck somewhere and it doesn't move to the end. So that's why you really have to test a few types of detection pad. Okay, this is my suggestion. This is my advice. If you buy too many, the same set, then if it doesn't work for you, so it's such a waste. So I think it's best to sample some different types of membrane pad, and if it works for you, then it's good. Yeah. So because it needs to capture the bioreceptor, but if you have a good bioreceptor, means very selective, then you're not going to be worried so much about the non-specific binding. As I mentioned, the capillary flow time is the most important for you to look on which type from the suppliers, okay, which type is good. So you have to do some work, to some optimization. It has to be optimized. So high capillary result in longer interaction, therefore better sensitivity. If it is too fast, the capture antibody or the capture bioreceptor might not be able to capture the antigen. So commonly used is nitrocellulose. This one has to be optimized. So in order to capture, means that to put the bioreceptor on top of the test line and the control line, these are very important things to consider. Of course, again, the buffer to maintain the right pH, the right ionic strength in order to put the bioreceptor. Okay, and stabilizers. Okay, because as I mentioned before, it's a wet and dry condition. So these stabilizers will help to maintain the structure of the bioreceptor, regardless if it is DNA, aptamers, or antibody. So this one will help to maintain the structure. So this is actually on the same detection pad, okay, the test line or the control line. So everything has to be there. Okay, alcohol for faster drying time, as well as also to stabilize. Okay. So again, the capture bioreceptor concentration will determine the signal achieved, of course. Okay. Sometimes because we know antibody is very expensive, we tend to put less, but it is suggested to use 1 milligram, more than 1 milligram per mil for sandwich non-competitive assay. Okay. If you are using a dispenser, machines means that you have to control the flow rate and the speed of the dispenser, position and size of the test line. This one you can read in figure 4, page 12, yeah, in the nature protocol section, page 12, figure 4. So you can see the diagrams of how the pictures being the pictures of how the process of sample pad, membrane pad, conjugated pads, how does it look? For those who are familiar, then it's okay. For those who have not come across the process, then this is how it looks. We have experienced two different types of fabrication. Manual, we did manually before, and also we have the dispensing system. Yeah, so of course the dispensing system is more automated and standardized compared to when we did the manual process. Okay, this one for those joining tomorrow, you can ask Dr. Fatin on the flow rates, on the speed, yeah? Because this is not something that you can just decide on one value. You have to do some optimization as well. Of course, this is the absorbent pad, so sometimes it is called wicking pad. Okay, at the end of the lateral flows, the strips, it is very important because it will stop the movement of the liquid, and it will also stop the back wash. I think we don't want the liquid to go back here, okay, because that will destroy whatever process that we are doing. So the absorbent pad or the wicking pad, the wick pad is very important as well. All right. So the whole process of this, what I have discussed just now, is you can view in figure 4, page 12. Okay. Of course, once you have prepared all this, it need to be mounted on the solid support. Okay. Right. And then after all the drying process, you can cut and put it in a cassette. Okay. Cassette here, I think quite common. If you buy the test strip, it is already in a cassette. Cassette ni tempat letak je lah, tak ada apa pun. So the last thing is to think about the storage. Okay, when your lateral flow is ready, readily fabricated, it has to be stored properly inside a sealed bag with desiccants. Okay, because it has to maintain in dry condition. And of course, because of the usage of a bioreceptor, you have to make sure it is cold condition. I think it also works if you just put it in room temperature. But if it is too hot, like you left it in the hot car, it might destroy. It might be destroyed. As well as you have to think about, you have to study on the lifetime, how long it expired, for example. That's something that you have to think if you want to commercialize this type of test. Okay, box 2, page 17. is how the assay, means the testing of the lateral flow. So it can, of course, commonly is qualitative, it's a yes or no. commonly used for lateral flow is yes or no. It can be semi-quantitative. Okay, so it's yeah. The Nature Protocol paper give you a very detailed procedure of how to use lateral flow assay. But of course, if you want to commercialize this one, we have to simplify this, okay? But they give you the critical steps, so you have to be careful on how to, whether you want to skip which is which. And the last part of the box 2, page 20, they give you the example of quantitative evaluation. So Dr. Diana asked just now, okay, so I think what you need to have is, of course, a reader. And most, I think, quite common now, they use smartphone, right, to capture, to take the picture of the test line as well as the control line together, probably. And you need to have an apps. We have before this, we have developed an apps with some other researchers. So they are not, I mean, there has to be someone else helping us from computer science, from AI people who can help develop the apps. Because we are chemists, I cannot explain on how to develop the apps, but this is doable. Means that they can do quantitative analysis by looking at the intensity of the lines produced in the test lines. It's doable, but it requires a lot of samples for the apps to be developed. Okay, if you ask, because they are going to train the AI. For example, this one, the concentration is 1 ppm or 2 ppm. It requires a lot of samples to develop the apps. Yeah. So if the apps is only just yes or no as well, it might be because sometimes we have difficulty in reading the strips because the line is very faint. It's very, so you're not sure it's positive or you're not sure it's negative. So that's going to be a very simple apps. Okay. But if it is for quantitative, you have to work with the engineer. You have to work with those E&E, electrical and electronic people, as well as AI people. So that one is beyond my capability to explain. And this one, apart from single analyte or single target, you can also use lateral flow for multiple targets. Okay, so you can do several targets by introducing several bioreceptors. So we have done this, 2 bioreceptors, okay, 2 bioreceptors, but we have used electrochemical techniques. We have not done 2 bioreceptors for lateral flow. So, but it's doable. It's doable. So multiplex reading is very important because sometimes you do not want to miss. For example, we are working with tuberculosis. So there are a few biomarkers that we can detect. So imagine is human. Sometimes they release one biomarker, sometimes they release multiple biomarkers. Probably the first biomarker is less than the second biomarker. So if we have more bioreceptors on top of the lateral flow, means that we can capture whatever secreted by the patient. Okay, so that is why multiplex is good, okay, because we do not want to miss any biomarkers released by the infected patient. So assay evaluation, you can also read table 4, page 21 until 22. As I mentioned, there's a lot of problem with lateral flow. So it's advisable for you to read 21 until 22. Okay, what happened if you don't get the reading? What happened if it's stuck? So very important to read through. We have experienced a lot of this, and we have thrown away a lot of strips because it doesn't work, especially when it got stuck. Yeah? So please do read this table 4, page 21 until 22. Okay, application. Some of applications I will discuss. Some applications, it does not cover all. Okay, but I find it interesting because it's a different principle. So what I would like to point out here is the usage of aptamer. So what we find is, based on our experience, the usage of aptamer saves us a lot, because aptamer is much more cheaper than antibody. I think for those who use antibody, we buy because we don't produce ourselves, then you know how much it is. I think for Mardi, they produce themselves, so it's quite, of course, cost effective. So that depends on you. But these are the biggest difference when you are using aptamer versus antibody. But the issue is how good your aptamer is. How good your aptamer is, how selective it is. That is also something that you need to question. But if you have a good aptamer, then the variation of the batches, okay, because we have experience when we buy antibody, it differs, the performance differs from batch to batch. Okay, it's expensive, and then yet it has different performance. So you can imagine how bad the thing will be. We have bought so expensive, and then it doesn't work because of the batch to batch variations. For aptamer, because it's chemically synthesized. So Dr. Azura's assembly of the FLA of the figure 4, page 12, itu rujuk kat mana? Dekat Nature Protocol. Dr. Fatin ada bagi sekali dalam, ada kan yang dalam email, ada bagi sekali. Ada ya? Okay, that's very important. Yang ni memang very comprehensive. Alright. Okay. Okay. So, but as long as you have a good aptamer, that is fine. Right. So this is an example. Why I'm using this example, because the person is in UKM. So she has done this before in her PhD, Dr. Hanun. So if you wish, probably you can communicate with her. I also put the paper here. Okay. She has done aptamer-based lateral flow. You can see that it's not much different. Okay. You still need to have control line and test line and control line. So for the test line, you're using one set of aptamer that is specific towards the target. And for the control line, you're using complementary aptamer. So if you refer to this paper, okay, if you have time, you refer to this paper, I can see that this one longer. And for some reason, I don't know. Okay. So I assume that because control line has to work, has to capture, just like antibody based lateral flow, where we put a common antibody set for the control line, as long as it can capture. So to make sure that the lateral flow works. Okay. But for the test line, of course, you need to have a very specific aptamer to make sure that you capture the right target. Okay. So as long as the rest are the same, the rest are similar, the sample pad, the conjugate pad, and the process of attaching aptamer to GO nanoparticles, okay, here, so aptamer and GO, that one you have to study the chemistry. As I mentioned, it is not in this workshop how to immobilize GO nanoparticles to the bioreceptor. It's not covered here. But yeah, if you wish, we can discuss it outside the class. Okay, it's very important. It's very crucial of how to the chemistry of how attaching the aptamer to the nanomaterial or nanoparticles. Some of you might be very good in this, so I'll leave it to you. And this is another example of using aptamer. And I have given the the link for you to explore. Okay, and but I would like to point out here, what's the difference? This is lateral. This is like a dipstick process. Yeah, you can see that this is the vials. So instead of having it lateral, we can put the strip vertical. Okay, so you can see that instead of dropping the samples, here we drop the samples, right? We can dip the strips in the vials, small vials, and the same process will happen, means that the sample will pass through the conjugated pad, the sample pad first, and then meet the conjugated nanoparticles. And this is also, I mean, the whole other things are similar, but the way that you introduce the samples, yeah, is usually we call it dipstick. Okay, this, of course, there are advantages. You might have more samples, okay, introduced, because if you do this, it can only receive a small amount. Yeah? You cannot like flood the sample pad. But if you choose this, then you might be able to introduce more samples. Yeah. So this is competitive and non-competitive assays, yeah, using aptamer. Okay, Dr. Diana asked about strip reader. There are portable strip reader that you can buy. You don't have to make one for yourself. You can buy. It's a simple colorimetric base. Okay, so whether it's quantitative or qualitative, I'm not sure, Dr. Diana, but there is a commercially based reader that you can buy. Okay, I mentioned about multiplexing. Multiplexing means that you can detect more than one target at one time. So you can see here, like influenza. Okay, we have many types of influenza. So we can have multiple bioreceptors, different types of bioreceptors on top of the same lateral flow or the same strip. Yeah. So when it goes from sample to here, so the bioreceptors will capture the specific target that match its each bioreceptors, each biomarker. Okay, So it is possible, yeah. So you can refer to this paper. There's a lot of latest paper as well. And I also provided you with this paper, integration of nanomaterial towards next generation. As I mentioned, we are so used to gold, but there are so many other nanoparticles that you can use and will give you a better results. So here there are also examples of using nano diamond, and it is fluorescent based. Based on the reading, it has much more, in terms of sensitivity wise, it is greater. Okay, so instead of just gold, we can use other nanoparticles. And if it is gold, for example, okay, here still using gold, but we can enhance the gold reading, the color of the gold, with addition of silver nanoparticles. Yeah, here, low sensitivity. We can enhance, means that it's a combination of gold nanoparticles added with Ag nanoparticles. So that, I mean, you can read the process in this paper. You can read this paper. There is one. So by having an intense color, the sensitivity of the lateral flow can be increased, because lateral flow is known for being not sensitive. Okay, but we can do the iteration or the improved sensitivity by adding this reagent called silver and some reducing agent to increase the color. So this is something that you can do. So this is a process. Okay, you can, I hope that you can read through the paper. Okay, this is the process, the silver staining, okay, applying to the gold. So the silver enhancement reagents are released from and are reduced to metallic silver around the gold. This is how it enhances the color. So the darkening of the TL here is the troponin. A tenfold sensitivity enhancement. So the use of silver staining is the keyword here, yeah, where it can increase the intensity of the color, so that it can detect much less lower concentration. Yeah. The other one is, okay, this one. All right. That. Okay. The use of magnetic nanoparticles, carbon nanotubes. Okay, instead of just using naked eye. Okay, so we can use fluorescence, Raman scattering reader, magnetic, okay, thermal reader, electrochemical reader. So means that you can put the reader here instead of just colorimetric, naked eye. We can use so many other types of readout signal to enhance the sensitivity, because this is something that we have to think about in future. If it is just for rapid test, screening is fine. But if it is meant for proper detection, then we have to think about this reader as well, apart from colorimetric. So this reader depends on what kind of nanoparticles that you are using. If it is fluorescent, then we have to use nanoparticle for fluorescent. If it is for Raman, something related to Raman scattering, electrochemical, then quite common. We can use also many types of nanoparticle because electrochemical is more robust compared to just colorimetric. And the last part, okay, is almost 12:30. We can also instead of just strips, normal strips that we use, we can embed the strips in another system. For example, here, we can embed the strips on mask, okay? So then you can imagine it can straight away detect the reading from the moisture of your, when you breathe in and out, it can detect straight away if there is positive line, then it is positive for certain disease, okay? So these are something that we can think about apart from normal strips. So strip is not just like normal strips, it can be incorporated together with other techniques like electrochemical, Raman, and also it can be in a form of wearable device, wearable things like mask. Yeah. So I think that's all. If there is any questions. Yeah, if there is any question from you. Okay, so tomorrow, Dr. Fatin will, for those joining the hands-on workshop, tomorrow you'll start at 9:30 and they will give you the location on how to get there. And so if there is no question, I hope I have managed to deliver some important things to you. And we also have sent you quite a number of important journals published, especially the nature protocols. Please download. If you miss the email or something, please communicate with Dr. Fatin to get the journals and as well as the slides. Okay, so for the last part, can I have like a picture with everybody? Everybody is so silent. Oh my God. Mm.